morning everybody. Today we shall talk about one of the most important chapter in immunology, that is transplantation immunology. Now coming to this transplantation, how do we define transplantation? When as a result of a disease or injury, an organ or tissue becomes irreparably damaged, or when an organ is congenitally defective or absent, a transplantation or grafting becomes necessary for restoring the function. That is, without this particular organ, the person might not be able to undergo his normal activities or might be even fatal to that particular person. In such circumstances, we go for transplantation. The individual from which the transplantation is obtained is known as the donor, and the individual on whom it is applied is called as the recipient. The first transplantation was first performed in our own country, that is in India, by one of the most important Ayurvedic surgeon, Sushruta Samhita, almost in 8000 BC. It was a skin transplantation on the nose by using the patient's own skin, and it was described in his books. The reason for rejection of transplantation till recently was thought to be active immunity. But in 1940, Medavar and his co-workers proved his immunological basis. Based on this particular transplantation, there are many number of Nobel Prizes which have been awarded to many number of scientists. In 1912, there was a person called Alexis Carrel from France who was given a Nobel Prize for his work on vascular suture and transplantation of blood vessels and organs. In 1960, Peter Brain Medavar was the one who discovered acquired immunological tolerance, and the graft rejection is an immune phenomenon. In 1950, he also induced immunological tolerance to skin grafts in mice by neonatal injection of allogenic cells. In 1990, Joseph E. Murray was also awarded Nobel Prize because he discovered that organ transplantation in treatment of many human diseases. In 1954, he also performed the first successful human kidney transplant between two twins. Transplants are possible in unrelated people if drugs were taken to suppress the immune reaction. In 1980, George Snell and Dean Dosset, these persons were given Nobel Prize for discoveries concerning genetically determined structures on the cell surface that regulate immunological reactions, that is H genes or histocompatibility genes and human transplantation antigens. In 1988, Gertrude B. Ilion and George H. Hutchinson, they were given a Nobel Prize for discovering one of the most important drugs to prevent organ rejection, that is immunosuppressive drugs. These days, we can attribute successful transplantations because of the discovery of this particular drugs. Today, kidney, pancreas, heart, lung, liver, bone marrow, cornea transplantations are performed among non-identical individuals with ever-increasing frequency and success. Now coming to the classification of transplants. Based on the organ or tissue transplanted, they are mostly classified as kidney, skin, liver and heart transplants, etc. Based on the anatomical site of origin, the transplant, and the site of placement or grafts, they are mostly classified as orthotropic and heterotropic transplantations. Transplantations may be of fresh tissues or organs or of stored ones. Transplant may be living or dead materials. Live grafts such as heart, kidneys, they are expected to function and perform the same function as they were performing in another individual. These are also called as vital grafts. Then we have non-living transplants like bone, artery, which merely perform a type of scaffolding or some type of support on which the new tissue is laid by the recipient. These are called as static or structural grafts. Now coming to the classification of grafts based on genetic relationships. Autologous grafts are otherwise called as autografts. That is, grafts transplanted from one part of the body to other part in the same individual. For example, in case of burn injuries, so the skin from the thighs is placed on the face, or skin or from other parts where the scarring is done, or sometimes in case there is a breakage of bone, especially on the arm, the ribs are removed and they are placed. That is, there is no any type of rejection that can take place. Then we have syngenic grafts, are also called as isografts. Grafts transplanted between two genetically identical individuals of the same species. That is, it can be monozygotic twins or dizygotic twins. Then we have allogenic grafts or allografts. Graft transplanted between two genetically different individuals of the same species. So most of the transplantations mostly occur in this particular area, which are called as allografts, where there are many chances of even rejection taking place. The new concept which might come in further future is xenogenic grafts, or otherwise called as xenografts. That is, graft transplanted between individuals of different species. So in case we are successful in xenografts, then I think most of the people can survive without only depending on the cadaver or any other transplantations. So this is a small diagram which shows you. So autografts, that is within an individual, then isografts between two identical twins, then we have allografts from genetically identical species, and xenografts from different species. Now, one of the most important rejection that takes place in grafts is graft rejection. So we have specificity, immune memory, and in graft rejection we have first set rejection and second set rejection. Now, when a skin graft from an animal is applied on a genetically unrelated animal of the same species, the graft first appears to be accepted initially. The graft seems to be morphologically and functionally active the first 2 to 3 days. So there is no inflammation, no necrosis, anything during the first 2 days. But on the 4th day, inflammation might set in and the graft is invaded by lymphocytes and macrophages. Then slowly ischemic necrosis sets in. The graft assumes a scab-like appearance and slogs off on the 1st day, 10th day. This rejection is also called as first set response. Now, if the animal or if an individual has rejected the graft by the first set of response, another graft from the same donor is applied, it will be rejected in an accelerated fashion. Necrosis sets in very early and within 6th day the graft gets slogged off. This is called as the second set response. So as you can see here, first and second set allograft rejection. So here we have a donor strain and this has what you call the skin from this particular has been placed on the recipient here, and there is a rejection after 3rd day, and by the 10th day that particular skin graft has been completely slogged off. This is called as first set rejection. Scientists thought that because it has rejected in the first phase, maybe it might accept it in the next phase. So they again continued with the same experiment. From the same donor they again placed it on another place, but here now the graft was rejected at a very faster rate, and this is called as second set response. They thought maybe skin was rejecting it, so they placed another type of organ. Even there also this particular mice rejected the graft in the second set response also. Now, what is the mechanism of this allograft rejection? An allograft will be accepted if the animal is rendered immunologically tolerant. So if suitable living cells are injected into fetal or neonatal animals of another inbred strain, when they grow up they will accept grafts from the former animals. As we were discussing in MHC, it is prior to birth, our T cells will recognize which are self and non-self. So by injecting such type of cells, we are making the immune system to consider them as self. In such cases, the graft will not be rejected. This is due to immunological tolerance as a result of contact with them during the embryonic life. The tolerance can be abolished by injecting lymphocytes from a non-tolerant animal. This method of transferring immunity by means of lymphocytes is also called as adaptive immunization. Allograft immunity is a generalized response directed against all antigens of the donor. A recipient who is sensitized by skin graft will also, by the second set of response, will also reject not only another skin graft but any other organ or tissue graft from the same donor. So as you can see here, these are some of the pictures which show the rejection process. So here the cornea rejection, as you can see, it has the whole of the cornea has been rejected after 10 days of the transplantation. Next. So here also you can see it's a skin graft. So you can see the skin graft has also been rejected after 10 days. This is also one example of second set of response. So with this we can say that transplantation can take place between two individuals only if rejection process can be eradicated. And this rejection process can be eradicated by using some type of immunosuppressive drugs. So these days we are successful in this allograftic or allograft transplantation because of these immunosuppressive drugs. But before that, most of the transplantations we have to check whether that particular person, the compatibility factor has to be more than 75% in order for the graft to be accepted. Thank you one and all for this particular presentation.